So, welcome to the YouTube channel. This is our first video. I am Dr. Muthu, uh, Assistant Professor in the Department of Basic Science from the School of Sciences and Humanities in SR University, which is located at Warangal, Telangana. So, let me give a small introduction about myself. I am basically a bioinformatician, computational biologist. We are doing computational drug discovery and gene expression profiles and machine learning and artificial intelligence related works. So the start of this video, we are going to see molecular docking of protein and ligand. We have This is a hands-on tutorial of how to do AutoDock from the scratch, how to retrieve the protein molecule and how to retrieve the ligand, and from where we have to retrieve and what format we have to retrieve, and how to process that protein and ligand and make them suitable for AutoDock. Then we are going to do AutoDock tools to do the molecular docking calculations of protein and ligand. So let us start with the downloading page. So where we have to download AutoDock. So it is available in the website MG Tools, Center for Computational and Structural Biology. Go to the website and then you will find here downloads. Here, if you have a Linux, there are multiple platforms are available: Linux and Windows and Macs. So we are gonna take Windows system. It's a 8 MB setups. You have to download it. Before that, we'll create one small working directory in the desktop. So in desktop, we have to create a new folder. So make remember that the new folder should not contain any space. We will mention as AutoDock tutorial. Okay. So when you are clicking this directory, make sure that there will be no space in any one of these names. Okay. If the space comes, it will be an error during the docking running process. Okay, make sure there is no space in the namespace of any folder. Then okay, the downloading has completed already. So once it is complete, we have to install. So we have to accept terms and conditions, then yes, and we have to select the path where it's supposed to install, then installing process. Usually it will be installed in the C folder program files. Okay, so after this there are two important files needed to run the AutoDock. So I will give these two files in comment box or link in the YouTube. So in this working folder, these two files are important: autodoc4.exe and autogrid4.exe. These two files are important to run the AutoDock program on your computer. Still the installation is going on. Okay, before that we will start with the retrieval of protein molecule. So protein molecule usually available in the website of protein data bank, that is RCSB. So if you go to protein data bank, all the proteins contain usually a 4-letter code. So I am here typing a sample PDB ID, which is MEK protein. So how to select the protein? This is one of the protein called mitogen activated protein kinase MEK1. So once you have come to this space, we have to see on what method it has processed. It is called as X-ray There are three different types of methods available in this database: X-ray diffraction, NMR, and electron microscopy. So we have to choose always X-ray diffraction method because of the easy pre-processing steps. Then we have to check the resolution. Resolution supposed to be always less than 2 Armstrong, which is very better. Then we have to see all these values to determine the to select the protein structure. And also, if you come little bit down, there is the information of literature. So where this protein has published, the literature information of protein publication is also available here. Then if you come little down, there are subsets. Like if the protein contains different chains, those chains are mentioned here. So this is only one chain is present in the protein: dual specificity hydrogen activated protein kinase, which has chain A, and it has a sequence length of 350 amino acids, and it has two mutations. Usually the cancer protein which contains mutations, it will cause cancer. If it is in wild type, it won't cause cancer. But some proteins we are targeting wild type proteins. In some cases we are going to check with wild type, in some cases we are going to check with a mutated type, in some cases we are going to check with both the cases. Okay. Then if you are little comes down, it is small molecule is nothing but your ligand molecule or drug molecule, which is also available here. So once you cross check all these things, you have to go to download then pdb file format. Yeah, so everything you have to include in the working directory. So I'll paste it here, 3w8q. So this is our protein molecule. If your protein structure, for example your desired protein, is not available in this website, you have to go to uniprot database. There you have to retrieve the raw protein sequence, then you have to model it, then you have to build the model. From there you have to predict your protein 3D structure. Then you have to retrieve the PDB three-dimensional structure for utilizing in AutoDock. So these are all the same protein retrieved from different publications and different organisms. So based on your study objective, you can take your own protein, then model it. Okay. Then it is also installation completed. Yeah, fine. So here we have the protein structure. We have to inspect the protein structure first using the normal notepad or notepad plus plus or something like that. So for molecular docking calculations, we need only the protein structure. So we have to eliminate the het atoms. Het atoms are nothing but your protein may contain water molecules. This is HOH is water molecules, and then we have het atom called AGS. That is one more small molecule or drug molecule which is present associated with the protein structure. So we have to remove all these het atoms and water molecules with the protein. So then we have to save the structure. Then now we can start the AutoDock. Initially we have retrieved the protein structure. Then okay, AutoDock has installed. Okay. Then how to retrieve the ligand molecule? This is protein 3W8Q. We can rename it as protein.pdb. Okay. Then we need ligand molecule. Ligand molecule can retrieved from variety of sources. So one of the source called PubChem. We can also retrieved from Campbell. There are many databases are there. For example, drug bank is another database. NPAC, zinc database. There are plenty of databases are available. It is each database contains unique characteristics. For example, in drug bank you can find the approved drug, FDA approved drug, investigational drugs, all these things. In PubChem you can find so many molecules. It's a mixture of natural compounds, drug molecules, synthesized compounds, individual metal compounds, all these things will be available. Maybe due to slow internet connection, I will take another protein molecule. Let us wait. Why it is not working? So wait for a second. Yeah, yeah, we can continue. This is the PubChem website. Here we can, for example, aspirin, a drug name called as aspirin. So when you search your drug name, it will come results like this. So when you click on the first results, here we have 2D, 3D, and crystal structure. For molecular docking, we usually choose three-dimensional structure. Here we can download the coordinates in the form of SDF. SDF is a structure defined file. We can rename it as aspirin. Then as usual, we will move to the our working folder of AutoDock. Yeah. So now we have protein structure and ligand structure in the form of SDF. So what we have to do is protein is PDB. It is in the correct format which can be readable in the AutoDock. But the aspirin.sdf which is not readable by AutoDock. So we have to convert this aspirin.sdf into the AutoDock suitable format. That means PDB format. For that, I'll use Open Babel to convert this SDF into the PDB molecule. So O Babel hyphen I. The input format is SDF, so we are going to convert this aspirin SDF. And output format is PDB, so we are going to take aspirin.pdb. Gen 3D is nothing but it will generate the 3D molecules. So yeah, 3D structure and PDB structure form. So you can find the PDB file here, aspirin PDB converter. So then now we can proceed with the AutoDock. So once you click AutoDock Tools, it will open like this. Then one small window will open. You should wait. There are two different ways to do AutoDock calculations. So in AutoDock tools, you can do one by one docking. Sorry, one versus one. You can dock one protein with one ligand molecule at a time. So there are options to dock multiple ligands with a single protein molecule using AutoDock V now. Okay, here, so the first step of AutoDock is to set the working directory. Go to the file and preference, then set. So in here in startup directory, we have to, so where is our working directory? We will close other files. Be confused. So for example, if it is your working directory, you have to select. The docking process has completed now. So we can click refresh and we can visualize the log file. Here, when you go to the last, the docking is finished. So 10 by 10, 10 out of 10 points has completed. So where we can find the docking score? Here in this file, when you scroll down, there is a table called histogram, or else you can type histogram, clustering histogram. Here we can find the 10 different docking score for 10 different ligand poses. So the lowest binding energy is 4 minus 4.31. So it is the top pose. Usually we will take the top one binding pose which has the highest binding energy. The binding energy is supposed to be always in the negative, and higher the negative is better, like minus 4, minus 5, minus 10. Minus 10 is 2 better than minus 4. So we have to select based on this. Then have to close this file. How we can retrieve the complex files, protein ligand complex files? So here, analyze, go to the docking again. Once you open doc.dlg file will come. Then again analyze. Already the macromolecule is present, so we can select the macromolecule. Then confirmations play. So once you click confirmations play, there is a small box will appear like this. So when you are moving like this, there is one ligand. See, here is the ligand. It will move now here. fourth pose, fifth pose, then sixth pose. So since our first conformational pose which achieved the highest binding energy, so we can set it as 1. Then that's the symbol, the like and symbol, so we have to click this, then we have to click right complex. So here complex dot pdbqt or pdb, we have to save like this. So the file will be saved in this folder complex.pdbqt. Yes. So once the file has saved, so we have to convert this into the pdb file for better visualization, because pdbqt is the file which is commonly autodock based file. It can be visualized in all the platforms, so we have to visualize to have to convert it. So obable hyphen ipdbqt, so complex dot pdbqt, and then output format we need as a pdb file, then complex dot pdb. So it will generate the warning because of the different number of poses. So once, see all are warning, there is no error occur, so we don't need to worry. Fine. File product start pdbqt. When you click open, it will ask for the ligand. You have to click ligand here. So this is your two-dimensional interaction diagram. So this is our ligand called as aspirin. This red color molecule is called as oxygens. So here we have amino acids. These the arc-like structures are also called as hydrophobic amino acid residues. Then here lysine 59, which has interacted with the two oxygen atom of aspirin with the hydrogen bond distance of 2.57 Armstrong and 2.96 Armstrong. So less distance indicates the higher binding affinity. Also the number of hydrogen bond, if more number of hydrogen bonds is better binding interaction between your ligand and your protein molecule. So from here we can take the screenshot or save the images. So this is the 2D visualization of your protein ligand interaction. Okay. Yeah, fine. So that's all from for this video. So if you have any queries or comments, you can mention it in the chat box. We will rectify it as soon as possible. Or if you find any flaws in these videos, you can mention also. Please likes and share and subscribe. Thank you very much.